Welcome back to part 2 of week 2. Today we are going to talk about hormones and what types of hormones there are, how they're transported, things like that. All right. Okay, so functions of hormones. First of all, these are just general overview functions. Hormones have different functions in terms of where they're released and what they target. But ultimately, what we're going to see is we're going to see hormones that control chemical reaction rates, maybe have an influence on an enzyme. We have some hormones that aid in transporting substances through membranes. We have some that regulate metabolism, water and electrolyte balance, blood pressure, obviously reproductive function, and then growth and development. So you're going to see some hormones maybe that just have one of these functions, but some that maybe have more than one. So target tissues. So remember that a target cell has a specific receptor for the hormone, and that hormone can't affect that cell unless that target cell has the correct receptor for that hormone to bind to. And some target cells are more sensitive to some hormones than others. And it does that through the number of receptors that are available for binding. So you can imagine that if you have 4 receptors and 4 molecules of hormone traveling through the bloodstream, all 4 of those hormone molecules all have an equal opportunity to bind to one of those 4 receptors. But let's say that you have 4 receptors, but you have over 20 hormone molecules. Of those 20, only 4 of those molecules can actually bind to those receptors. And so in that sense, maybe the body isn't as sensitive to that hormone than it should be. The more receptors that it has, the more sensitive it is to the hormone. So when we talk about hormone regulation, we're talking about upregulation and downregulation. Upregulation is when there's an increase in receptors on the cell. So let's just say that we have those 4 receptors and 20 molecules, but now our body has made more receptors. And now we maybe have 15 receptors to 20 molecules of hormone. So that just allows that cell to become more sensitive to the amount of hormone that's being released. All that means is that at homeostasis, maybe we have these 4 receptors and maybe that's enough. But in terms of more hormone being produced for whatever reason, now we need receptors to accommodate this overproduction of hormone. So then your body can go into these mechanisms to make more receptors and therefore make your cells more sensitive to that hormone and have them bind up more. That's upregulation. Downregulation is going to be the opposite. So let's say that we have 15 receptors, and now our hormone level has decreased to about 4 molecules, back to homeostasis, right? But we have 15 receptors. So definitely there's enough for all 4 of those molecules to bind to, but there's also going to be 11 extra receptors. And so what happens is that those receptors are going to become less sensitive to that hormone. And so we're going to decrease the number of receptors that are available for binding and decrease it to accommodate how much hormone is actually in the bloodstreams. And that's downregulation. So upregulation and downregulation is basically the body's ability to control the number of receptors on a target cell to accommodate how much hormone is being released by the body. And it's all about homeostasis. The more hormone that is being produced and released, the more receptors you need, and vice versa. Okay, so here's just a good image of this. So upregulation, here we have a ton of hormone here in the bloodstream, but we only have 3 receptors that are available. And so what our body is going to do is upregulate. And so we have multiple receptors that are available for all of that hormone to bind to. And therefore, that's going to increase your sensitivity to that hormone. Downregulation is the opposite. So we have a ton of receptors, but we only need a few. And so we're going to eliminate some of those receptors, recycle them, so that now we have 4 receptors. And so your response is much more diminished. The more receptors that you have, the greater response you're going to have, because the more hormone that's going to bind and send their chemical messages through to the target cell. The less number of receptors you have, the less robust response you're going to have to that hormone. Okay, so here's a good example of type 2 diabetes. Type 2 diabetes is an insulin insensitivity. So people who are diagnosed with type 2 diabetes have chronic increased blood glucose levels. And when you have lots of blood glucose, your body produces a lot of insulin. Insulin is the hormone that takes glucose out of the bloodstream, stores it as something called glycogen in the liver out of the bloodstream to bring those blood glucose levels back down to normal. Okay, so if you have this chronic blood glucose level that's constantly high because of maybe your diet, right? Let's just say you eat a lot of carbohydrate heavy meals, you're going to produce more insulin to combat that. But what's going to happen is that more insulin that you produce, okay, the number of receptors are going to go down because the normal amount of insulin is here and you're producing this much insulin, okay, and those receptors need to be here. And so what's going to happen is those number of receptors are going to decrease because you have so much insulin, and so you're going to become less sensitive to that insulin over time. So that is an example of downregulation. So here, high levels of insulin normally have high levels of receptors, but homeostasis says that we need to take those receptors back down to a smaller number. And so you have a smaller number of receptors. Well, you still have a high number of insulin molecules circulating in the bloodstream, but only a few of them can bind. And so that's going to cause a diminished response of that insulin and cause the individual to become less susceptible or less sensitive to the insulin. And so that is why people who are type 2 diabetic need to take supplemental insulin. Another example would be pregnancy and labor. So this is an example of upregulation. So women in the late stages of pregnancy, their uterine cells produce more receptors that are sensitive to a hormone called oxytocin, and oxytocin stimulates uterine contractions during labor. And the more receptors that you have, the more oxytocin can bind. And so what your body does at late stages of pregnancy is produce a lot of those receptors in preparation for the extensive amount of oxytocin that's going to be released upon childbirth and labor. And so that way your receptors can become more sensitive to that oxytocin. So we have lots of hormones, but they can be pretty much categorized into a few different types. The only types that we're going to talk about are steroid and non-steroid. There are also some other ones like prostaglandins and things like that we're not going to talk about. But the two main ones that I do want to cover are steroid hormones and non-steroid hormones. So steroids are synthesized from a molecule called cholesterol. So these are going to be like, excuse me, your sex hormones, your corticosteroids, your calcitriol, and all of these you'll notice usually will end in all or own or ogen. Okay, so their endings are going to indicate that they're steroids. And the difference between steroids and non-steroids are they're going to be their chemical basis. So this is synthesized from cholesterol. Now, the thing that we notice about steroid hormones is that they are slower acting and longer lived. So, for example, if somebody if somebody takes testosterone injections, they're not going to feel the effect right away because that testosterone takes a while to get into the body cells and do what it needs to do. And let's talk about that. So steroid hormones are poorly soluble in water because they are from cholesterol molecules, right? Fatty acids or lipids. So they are weakly bound to plasma proteins to help them get through water. If they're poorly soluble in water, think that they cannot be transported in water by themselves. They need something to help them. And so there are plasma proteins circulating in your bloodstream that bind up these steroid hormones to help carry them through the bloodstream to where they need to go. That can be beneficial because they are bound to plasma proteins, they can circulate for longer periods of time in the body. The other thing about steroid hormones is that these are soluble through cell membranes. So remember, cell membranes are made up of phospholipids. So cholesterol is also a chain of lipids. And so these steroid hormones can freely cross that plasma membrane, no problem. And once they're in the cell, they're going to bind up a receptor. The receptor is actually located in the cell rather than on the cell surface for steroid hormones. And it's going to enter the nucleus of the cell and bind up DNA sequences, form mRNA sequences, and then translate into proteins that are necessary for whatever action that hormone is intended for. And so if you remember central dogma, right, DNA is transcribed into RNA, which is translated into protein. And once this hormone receptor complex gets into the nucleus, it binds up DNA sequences, forms a recipe or an mRNA sequence, and then that's going to translate into protein, most typically activating or repressing genes. But also we can make maybe more proteins such as a sodium potassium pump. For example, if our fluid balance is really out of whack, we can maybe make more pumps to help return that level to homeostasis. So we're going to get over some examples here. So here's a good image from your book on how steroid molecules get into the cells. So steroid hormone molecule is out here in the bloodstream traveling along, right? And he should be bound up to something, but let's just say that that carrier protein unbound him by this point. And because he's a lipid, he can cross this phospholipid bilayer freely, okay? And he's going to bind up, or he's going to enter the nucleus rather, and bind up a receptor that's in the nucleus here, okay? Once that hormone receptor complex is initiated, then it can bind up to DNA and create an mRNA sequence. And we know that mRNA leaves the nucleus and goes out into the cytoplasm of the cell, and then ribosomes translate that into different proteins. And in this case, this one is forming a new protein molecule, whether that's an enzyme to complete a chemical reaction, whether that's a pump of some sort to maintain fluid balance. It could be a variety of different types of proteins. But essentially what it's going to do is upregulate or downregulate a genetic sequence in order to complete a cellular change. Okay, so an example of this would be in the kidneys. So for example, aldosterone ends in ONE, right? That's a good example of a steroid molecule. And let's just say that our stimulus in the body is that our blood pressure is dropping below normal. Okay, so when blood pressure drops below normal, our adrenal cortex releases a hormone called aldosterone. And aldosterone is responsible for telling the kidneys to make more sodium and potassium pumps. And from the foundational concept chapter, we now know that sodium and potassium pumps move sodium and potassium back outside the cell or against their concentration gradient, outside or in, depending on if they're sodium or potassium. But basically, they're moving these ions against their concentration gradient. So naturally, this is a nephron in the glomerulus in the kidney, and we're going to get to this more when we get to the urinary system. We're going to go over this all again. But naturally, what happens here as blood flows into the kidney, sodium leaves the bloodstream. Now, think about this. If sodium's leaving the bloodstream, what other molecule is also following sodium? About water, right? Water follows solutes. So if sodium's leaving and going into this tubular system, which actually is how urine is formed, water's following, that diminishes the amount of water that's actually in the bloodstream. The less water you have in the bloodstream, the less blood volume that you have, okay? And the less blood volume that you have, the less your blood pressure, okay? So how do we fix this problem? We want to reabsorb that sodium that went out again. It went out with its concentration gradient, and we're going to try and pull it back in against its concentration gradient. So we're going to do that with sodium and potassium pumps. So what aldosterone does is tells the kidneys to make more sodium potassium pumps, excuse me, so that we can reabsorb the sodium that was filtered out. And when we reabsorb that sodium back into our bloodstream, water is going to follow it. That's going to increase our blood volume, so the amount of blood that we have in these vessels. And as we increase volume, we're also increasing pressure. Okay, hang on to that concept. We are going to go over that a lot more when we get to blood vessels, blood pressure, and cardiovascular system. Now, different from non-steroid hormones. So your non-steroid hormones can be categorized into different categories based upon their chemical makeup. We're not going to get into the chemistry so much, but they're mainly categorized into molecules called amines, proteins, or peptides. Amines are derivatives of the amino acid tyrosine. Proteins are long folded chains of amino acids. Hopefully you know that already. Peptides are short chains of amino acids. So a peptide is basically just a short protein. And amine is the shortest of all of them. So amines, we're talking about epinephrine, norepinephrine, thyroxine from the thyroid, melatonin. Long proteins are going to be like parathyroid hormone, all of the anterior pituitary hormones. Shorter chains of amino acid are going to be the posterior pituitary and the hypothalamic. So, you know, I'm not going to make you know the structure of all of these different types of hormones, but just know that the non-steroid hormones, there's a huge variety of them, and they're all categorized based upon their chemical makeup. Now, different from steroid hormones, non-steroid hormones can freely travel through the bloodstream. They don't need a carrier protein to carry them through the bloodstream. They're faster acting, therefore, and short-lived. So if you think about that for a second, think about epinephrine. Okay, epinephrine, if you're familiar with epinephrine, it raises your heart rate, raises your blood pressure, all those types of things, but it's very short-lived. Okay, so faster acting and shorter lived, right? You can give epi for an allergic reaction and save a lot of people's lives, right? Now, the difference is that non-steroid hormones, okay, are not lipid based. And so they cannot cross the lipid membrane of its target cell by itself, okay? What it will do then is find a receptor on the surface. Okay, so before, the steroid hormone had the receptor in the nucleus or the cytoplasm, depending on what kind of steroid hormone you are. For non-steroid hormones, the receptor is on the cell surface because that hormone cannot enter the cell membrane through that lipid bilayer. And that binding of the receptor to the hormone is called a first messenger. Okay. Now, that signals inside the cell to trigger a cascade of second messenger processes. Okay, so this gets a little complicated, but I'm going to go through it step by step, and I would encourage you try to draw these out as well. So once that hormone binds the receptor on the cell surface, inside the cell, when that happens, that signals this thing called a G protein to bind to that hormone receptor complex as well. He's the second he's like a little messenger inside the cell that pulls the signal from the outside the cell to the inside of the cell. I'll show you a picture in a second. The G protein will then bind something called adenylate cyclase, which removes 2-phosphates from ATP and connects an oxygen. And all that means is that it forms another molecule called cyclic AMP. Okay, then cyclic AMP is your second messenger. And what he's going to do is activate protein kinases. What those do is basically activate or deactivate proteins that are already made. So remember that with the steroid molecule, the goal was to provide the instructions in order to make the proteins that are needed, right? That takes a lot of time. That's why those are longer to activate, right? Those types of hormones. Non-steroid hormones, the proteins are already available. We just need to activate them. And we do that with an enzyme called protein kinase. So here's what this looks like, a little bit different from steroid hormones. So here's your hormone out here, your steroid hormone. And notice how here in the plasma membrane, we have this receptor. Okay, so that hormone is going to bind that receptor On the inside of the cell, there's a G protein connected to that receptor. And what that G protein is going to do is come over here to this adenylate cyclase, which is also in the membrane. And that adenylate cyclase is going to do a really complicated chemical reaction and basically form something called cyclic AMP. And then cyclic AMP is going to come over here and tell these protein kinases to start working, right? These enzymes, and that's going to activate different proteins to promote cellular changes of whatever it might be, right? So like epinephrine, for example, let's say this is epinephrine, and that binds up this, and then the G protein comes over here, forms adenylate cyclase, which forms cyclic AMP, which tells whatever protein is involved in increasing the heart rate, right, to start working and increase that heart rate. Okay, so the difference between steroid and non-steroid is that steroid molecules have to have something carrying them through the bloodstream, but they can freely enter the cell. Non-steroid molecules cannot freely enter the cell. They can travel through the bloodstream by themselves, but they need to bind a receptor out here, and that initiates this cascade of internal things. So I don't need you to know the specifics of all of these steps, but you should be able to write them out. Okay, like step 1, a non-steroid hormone binds a membrane receptor, right? Step 2, the G protein binds adenylate cyclase and forms cyclic AMP. Okay, number 3, cyclic AMP activates protein kinases, which then activate or deactivate proteins depending on what's going on. And that's really as simple as you need to make it. Okay. An example, so during a period of physical stress, we'll talk about stress yet this week. What our body wants to do is mobilize more glucose for energy, right? The more stress we're under, the more energy our body uses. And so we need to mobilize as much glucose as possible to be able to use for cellular energy. So what our body does is maybe release epinephrine. Okay, epinephrine is going to bind the receptor on the outside of the membrane and stimulate that intracellular cascade. And what cyclic AMP does is going to increase the activity of the enzyme or protein that breaks down liver glycogen into glucose. So glycogen is the storage molecule that glucose is stored in the liver. And so we can tap into those stores whenever glucose levels are low or whenever our body needs more energy. So we just break down that glycogen, basically unwrap it, and here we have mobilized glucose, right? And then that increases the amount of glucose circulation in our body. So this would be an example of non-steroid hormone action. Negative feedback, right? So maintaining those constant hormone levels in our blood, it's going to fluctuate constantly. How these are controlled, how our hormone secretions are controlled is in one of 3 ways. Okay, so we have the hypothalamus. That's kind of up here in the brain, kind of a main hub of the body in terms of endocrine. The hypothalamus senses the internal environment, what's going on. And the hypothalamus controls all of the anterior pituitary secretions. Okay, so this is the first really important concept that I need you to know, is that the hypothalamus is going to control the anterior pituitary. And then the anterior pituitary secretes hormones that act on some other glands. We'll get there in just a little bit. That's one way. The second way is that the nervous system stimulates glands on its own. So remember that I said the posterior pituitary was made up of axons, okay? And that's stimulated by the nervous system. So the posterior pituitary, for example, is not stimulated by the hypothalamus, but it's stimulated by the nervous system. Same with the adrenal medulla. Remember when we looked at the histology of the cortex and the medulla of the adrenal gland, the medulla was lighter. It's stimulated by the nervous system, right? The adrenal medulla is stimulated by the nervous system to release some hormones involved with increasing our fight or flight response. And the third way is that some glands just respond directly to their own internal environment changes. So they detect their own changes and respond accordingly. So the pancreas does its own self-detection of the environment. and is going to release glucagon or insulin, depending on if blood sugar levels are low or high, respectively. Okay, so either the hypothalamus is going to start the cascade, the nervous system is going to start the cascade, or the gland itself is just going to start and stop the cascade. So those are the three different ways that hormones are controlled and secreted. And it's all about negative feedback. So let's take the first example. We have the hypothalamus. The hypothalamus is always going to stimulate the anterior pituitary. And then the anterior pituitary is going to stimulate an endocrine gland somewhere else in the body, which is going to stimulate a target cell and do an action. Now, let's say that this hypothalamus is releasing a lot of hormone that tells the anterior pituitary gland to release a lot of hormone. This peripheral endocrine gland can say, okay, hold on, we need to stop, right? Slow it down. You need to quit giving me so much hormone to stimulate me. And I'm going to tell either the anterior pituitary to slow down, or I'm going to tell the hypothalamus to slow down and then re-regulate those levels. For the nervous system, the negative feedback comes from the action. So let's just say epinephrine, right? So let's say that the nervous system is stimulated in terms of, I don't know, fear. Okay. So fear, those messages are going to go down the nervous system to the adrenal medulla. The adrenal medulla is going to secrete epinephrine, which is going to increase heart rate, blood pressure, all of that. Once that heart rate gets too high, maybe, right? That's going to stimulate the nervous system to take it down a notch, right? Quit stimulating me, or heart rate's going way too high, or blood pressure's going too high, right? And that's going to cause another slew of problems. And then the self-detection of hormone levels, that's this third example. The action also regulates the stimulus here. So changing level of substance in a plasma, let's just say that your sodium's too low in your bloodstream, right? So if your sodium's too low in your bloodstream, maybe the posterior pituitary or the adrenal cortex is stimulated, and that targets the kidneys, and the kidneys are going to reabsorb that sodium and therefore water. Okay, but what happens if you've reabsorbed too much sodium or too much water? Now your blood pressure is going to rise too high. And so we have a lot of mechanisms intact there that are going to regulate that and tell, our adrenals or posterior pituitary to quit releasing the hormone that's telling me to do that, right? Because we want to maintain that state of homeostasis. So here's an example here of something that's too high or too low. So let's just say that our hormone levels get too high. The receptor is that the control mechanisms, whatever that might be that we just talked about, sense that change. And so now you're going to inhibit that gland, right? You're going to tell it to quit releasing so much hormone. And so then your levels will return back to normal. And then too low would be a similar response, except for we want to release more endocrine hormone. And just know that this is what negative feedback looks like. This is what homeostasis looks like. It's never a straight line, right? Let's say that your average concentration of hormone is here. So let's just say, for example, like the normal circulating epinephrine that's in the bloodstream is this number, whatever this number is. You're constantly going to have lower than that, higher than that, lower than that, higher than that, but ultimately it's going to fluctuate like this about that set point or that normal average level that we want. Okay, so just know that these endocrine glands are constantly being stimulated or inhibited to keep the range as close to normal as possible. And that's what negative feedback is. So what happens with athletes who dope steroids, right? So a very common anabolic steroid would be testosterone. The intent is to increase muscular strength. So yes, in young males, when they hit puberty, testosterone is necessary for muscle development. Now, adults take testosterone in order to enhance muscle performance, like bodybuilders, for example. The side effects of that, though, is having too much testosterone in your system was actually some pretty detrimental effects here. So we're going to increase the level of which you reach adulthood. You're going to have lessened height growth, early hair loss, maybe some breast development. Now, this comes from the idea that testosterone and estrogen have to be in analogous levels of each other, like they have to be kind of balanced. Well, if you're increasing your testosterone too much, now your estrogen is also going to increase. And estrogen in males at high levels cause feminine characteristics. And so that's where we kind of get that. Now in females, they might get a deeper voice, hair growth, irregular menses, things that we associate with biological males who have high levels of testosterone, right? What testosterone also does is cause an increase in LDL, which is the bad cholesterol, and a decrease in HDL, which is the good cholesterol. And therefore we have fatty deposits in the blood vessels, which is called atherosclerosis. And then you can be at risk for stroke and heart attack and things like that. There are some psychiatric side effects as well. So some people decide to supplement that with growth hormone in order to grow the muscles and make them stronger at the same time. But here's the question that I want you to kind of simmer on here for just a little bit. I'm going to come back to this in a future lecture. But how do you think injecting high levels of testosterone would affect a negative feedback mechanism of testosterone? And we're going to talk about the mechanism of testosterone release here in a future lecture. So I just want you to keep this in the back of your head. We talked about those negative feedback mechanisms, but why would high levels of circulating testosterone affect negative feedback? Okay, keep that in your mind. Another athletic problem is called blood doping, if you've ever heard of this. Essentially, the liver and kidneys are responsible for secreting a hormone that stimulates red blood cell production. And your red blood cells are responsible for carrying oxygen throughout the body as hemoglobin on molecules called hemoglobin. And so you can imagine if you're an athlete and you can somehow increase the oxygen carrying capacity of your body, that you maybe would have a higher aerobic endurance, right? Maybe runners or bicyclers or something like that. Well, what happens, the more red blood cells that you produce in your bloodstream, think of those as solutes, okay? So the more solutes that you have in your bloodstream, the thicker your blood becomes, right? Because now you have more solutes, but you don't have enough water to balance out that solute concentration. So it makes your blood thicker. And side effects of thick blood, heart attack, stroke, pulmonary embolism, possibly death, things of that nature. So that's the why, right? Because your blood is thicker because you have more cells circulating in your bloodstream. Okay, so that ends up lecture 2, part 2 of this series. In the next lecture, we're going to go through some actual hormone pathways from start to finish. So I will see you there.